back. So let's talk testing. The tests for monkeypox virus are going to be similar to the tests for most virus. The best way to detect this thing is by PCR. And you can do PCR on patient samples. You can do PCR on wastewater because people will often get an intestinal infection with monkeypox because, like I said, it's a promiscuous virus. Goes where it wants to, knocks on a lot of doors, gets into a lot of cells, causes a lot of problems. Quantitative PCR is what we're doing here. And so it involves two primers, a forward primer going this way, reverse primer going this way, which is actually the reverse complement of this sequence. And then in the middle, there's a thing called a probe. And so on one end of the probe, there will be a fluorescent group. On the other end of the probe, there will be a thing called a quenching group. And the quenching group will basically interact with the fluorescent group in such a way that the when light goes into the fluorescent group and the fluorescent group changes the wavelength and sends it back, because that's really all fluorescence is, then the quencher is going to grab that and it's going to radiate it out as something else. You're not going to see that light coming out. And so the quencher quenches. When a polymerase binds here to the forward primer, it will then start copying, and when it runs into the probe, it will start degrading the probe. And so it will separate the quencher and the fluorescent group. And so you'll end up with, quencher far away from fluorescent group, and then you start to be able to see the fluorescence. And this is how it works. We can then just detect the fluorescence, and from the amount of fluorescence, you can tell that you're doing PCR, that you got a positive hit, and you can tell how much you started with, because the more copies you have, the faster this sort of glow. People will often use either red or green fluorescent here. The faster that will accumulate, and it's a predictable rate, so it's easy. Yeah. Easy to study anyway. And so, yeah, you'll have two primers either side, and you'll have a probe in the middle. And that's quantitative PCR. There you go. All right. So, before 2022's big outbreak, the most common places where you would encounter monkeypox, or we should probably call it human monkeypox virus, because it doesn't grow in many monkeys outside the laboratory where people give it to the monkeys. So Nigeria, I believe, is the bulk of the cases of the clade B, or the less virulent clade. And the clade A, or more virulent clade, tends to be more in Central Africa, Democratic Republic of the Congo, and Central African Republic. Less so in Cameroon, or perhaps they have bad data from there. That does happen in some countries that are dealing with other bigger problems, frankly. Okay. The new strains of the virus, so these are some new genotypes of the virus from very early in the pandemic spread of 2022. They are related to some of these other strains. And if you look at the other strains, a lot of those come from the gray color, which is Nigeria. And so what you got here are viruses that are just possibly circulating or possibly jumping from animal to human in Nigeria. And honestly, I don't think the answer is completely known for most cases. And then it'll get out. Somebody will perhaps be in Nigeria and come out and visit family, or come back to the United States. And you get a little bit of spread sometimes, and then usually the trail goes dry. Something happened, and one of these Nigerian-like strains ends up jumping, and it ends up infecting tons of people and spreading out. And this is the point where we kind of notice that there has been a problem. But some of the early steps are still a little hard to be certain what happened, where. We know some of them, but not all of them. There are mysteries here. Okay. Now, this is interesting, because there are a lot of older people that have had the smallpox vaccine, and the smallpox vaccine will protect against monkeypox. But vaccines wear off. And so this is from an older paper where they look at people who have the virus. They look at people in their household that they are presumably breathing in their presence, potentially touching them. And yeah, they find that the attack rate for people living in the same house as an infected person is about 50%. And so this is being done at a time where there wasn't a lot of PCR screening. So it's 50% of the people in the house go on to develop symptoms. It may be all the people actually get infected. It is hard to tell. There's basically no data. So rather than speculate, we'll say at least 50% of the people in those households were getting infected. Several of those cases were in people who had been previously vaccinated for smallpox. So because of that, we essentially know that, yeah, shown down here in sort of cyan, several people, not many, but some, had been vaccinated and did get an infection, because vaccines are not perfect. They're good. They're a whole lot better than nothing, but they're not complete invulnerability like people mostly want them to be. And maybe in the future we can get better at it, but I think the weak component is the immune system, and we can't really do much to change that without some heavy genetic editing of people, and I don't think the world's ready for that just yet. And frankly, I don't know if we would know what to do if people, like the tools are there, but what do you do with the tools? That's a good question. All right, so how does this happen? These are some of the known hosts for human monkeypox virus. Many of them occur in Central Africa. These are the ones that are most likely to be associated with the animal to human transmission. But throughout the world, there have been cases of human to animal transmission, and sometimes then animal back to human transmission. There was a famous case with some prairie dogs in the U.S. getting infected and, yeah, causing very bad things to happen. So in addition to all these different animals in the native sort of area, you've got a lot of different creatures. So there's our prairie dogs down at the bottom that can be infected in other countries. Some of these are imported from Africa, but others are not. So, like I said, there's a lot more capacity for a virus like monkeypox to grow and spread than there is for some other viruses. Like smallpox, for example, was fairly restricted. It was never found in anything but a human, if I remember correctly. So, yeah, monkeypox is a bad dude. Yeah. All right. Speaking of those prairie dogs, when they were infected, where did the virus go? So people can now use prairie dogs as a model for infection because they're very readily infected with monkeypox virus. So it's a good way to study it. And with most viruses, what you see is that the virus will be a lot in certain tissues and very little in other tissues. What you get with monkeypox is just that the virus is everywhere. It's in fecal samples, it's in blood, it's in oral samples, it's in samples from the eye, ocular samples, and it's in nasal samples. So that's pretty much all the surfaces of the body, inside and out, that we're covering there. And there are actually a lot more tissues among these that the virus can infect. So yeah, fairly dangerous. Once it gets in, it can do a lot of bad things. There's basically nothing good that can happen from that point. Now let's talk about the vaccine. So the vaccine has been largely discontinued, at least the old version of the vaccine, because there were some complications. Now they're rare. They used to vaccinate babies, and in about 1 in a million cases, you would get unchecked spread, where the virus essentially just sort of grows outside the area where it was inoculated. The vaccine is live virus, normally live vaccinia virus. And the new version of the vaccine that works against monkeypox virus that's approved for it is a thing called a modified vaccinia virus. And we'll get into what that means in another slide, I think. But this is the procedure. You take a needle. You can use one of these that has 2 little bifurcations on it. You get a little drop of the virus in between those 2, and you mash it into somebody's arm. Mash, 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 mash. because you need to stir up some macrophages. You need to build a little bit of an immune response around there as you bring in those dendritic cells and macrophages. Those are the cells that can become infected, and they'll be more or less localized at the site. And because this is a mild version of the virus, then they will get infected. You'll make a nice immune response against it, and that immune response will then last for years and years afterward, although potentially not forever, as we're finding out with the monkeypox vaccinated people that got monkeypox anyway. And then the problem comes in that you get this lesion at the site. It's like a little pustule, fills up, and then it sort of collapses and scabs over. And once the scab falls off, at that point it's safe. But anytime up until then, there's actually live infectious virus with some number of membranes. Any number of membranes will work, for this weird virus. And if you rub it and then rub your eye incidentally, which people do all the time, even though they think they don't, then what you'll see is that, you can get ocular vaccinia, like so. And remember that your eyes are just a little part of your brain that sort of pokes out through two holes in the front of your head. And so if you're getting an eye infection, you actually have a brain infection. And brain infections can be serious. And so they can lead to other bad things. So that was the risk. That's why it's not widely used outside of the military, I believe, still uses it. But the new version of the vaccine is actually much safer to use. So, new version of the vaccine is called Modified Vaccinia Virus Ankara, as in the city in Turkey. I think the capital of Turkey. So it was attenuated in Germany back in the 1950s. You say, what is this brand new technology? No. This is your grandparents' technology, maybe a little before their time. What they did basically was to grow the virus over and over and over again in weird cells. So they grew it in chicken cells. From little chicken embryos. You basically take an egg that has a little embryo in it and you mince that embryo up and you let whatever cells are in there grow, and then you can infect those. Weird, barbaric, useful, and the way that this was done. And so after nearly 600 rounds of this crazy process, then they ended up with a virus. And they tested it out along the way, and they found out that this had some genetic defects. So it was missing certain areas. It had big deletions in some of those. It was still able to grow, but it was much less likely to cause disease because it couldn't complete its full replication cycle in human cells. It could complete it in chicken cells. It is adapted to the chicken cell. And for whatever reason, in the case of this virus, and this is not like a thing you can do every time and have it work, but in this virus, it got used to working with the chicken proteins because the virus is going to interact with host proteins to do a lot of things. And it actually lost many components that it needed to grow in a human cell and made many mistakes. So, it's happy. The other nice news here is that we have tecovirimat. And so here's the name, Tecovirimat. And tecovirimat is a drug that can be used to stop many different poxviruses. And so here you can see an EC50 value, like the effective concentration to knock out half of a virus. And I believe these are in micromolar. So these would be, sort of medium nanomolar EC50s, which would be good. Those are good EC50 values. But you can see it works about the same against a whole broad range of poxviruses, many of which can, under some condition, actually get into humans. So this is nice. Here's from the paper where they actually tested these antivirals, tecovirimat. And so they show that if you have no drug, that you will have lots of virus coming out. Basically, if you have drug, then the infection lasts a shorter time. So you get right down to here. So instead of having virus coming out for a long time, you treat, somewhere around here, and then the virus will go away faster, which is exactly what you want. Here's our placebo treatment up here. We get tons and tons of copies of the virus in the blood, about 10 to the 8th per mil, which is a lot. And then with the actual treatment, it goes down to the lower limit of detection, which is right around there, around 200 copies, very quickly. Total number of lesions is there are no lesions, and the animals they infected are essentially okay. So those were the experiments that led to this. This was also tested out in cynomolgus macaques, which is a type of monkey, if you like. And same thing. The virus went up. The virus came right down when they treated it. The virus went up, and the virus just sort of lingered when they didn't treat it. And the monkeys went down, 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 and then died when they didn't treat it, or stayed alive, put on weight, did nice primate things for a long period. Essentially no problem. So, nice, they work. How does it work? Well, tecovirimat, which is also called ST246, is an inhibitor of Rab9, among other things. And so this complex with Rab9 and TIP47 and P37 in it is going to intermediate between the late endosome and the sort of wrapping complex, where the virus is going to wrap itself up. The components of the virus come along, and essentially it is unable to do wrapping because you've interfered with the components of the cell that the virus was relying on to help it in the wrapping process. And so, it's really nice. A way to stop one of the stages of the virus replication cycle well enough that you can actually win. You can actually beat the virus. So, good job. Way to go. Yeah. Okay, and that's where we'll pause that section. And then we have one more section when you come back. All right, see you then.